Good day, learners. Welcome to the grade 11 life science lesson. My name is Shabnam Sugnandan. This lesson is brought to you by the Sci-Bono Discovery Center in collaboration with the Gauteng Department of Education. So today's learners, we're going to be focusing on photosynthesis, and the subtopic of it is going to be on the factors that affect the rate of photosynthesis. So what are the factors that are affecting the rate of photosynthesis? So learners, this topic is actually very abstract and relates to graphs, and you have to actually analyze all the data that is found in the graph in order for you to understand how certain factors affect the rate of photosynthesis. Before we proceed, let us just do a little recap of the previous lessons on photosynthesis. Remember, learners, that photosynthesis may seem like an unnecessary topic, but remember that without photosynthesis, life on Earth cannot exist. Okay, so we're going to look at photosynthesis, and you remember, just to recap, what were the raw materials that were involved in photosynthesis? And it was what? First of all, you need carbon dioxide. That is actually symbolic form, CO2. That is coming from the air. And where does it enter into the leaf? Remember, the leaf is the main photosynthetic organ. So the carbon dioxide must enter via the little openings called the stomata. So that's where the carbon dioxide that we exhale, that is given off by the fires, that CO2 cannot be too much in the air. And the reason why it cannot accumulate in the air is because it can form carbonic acid. But it's because of our plants that the amount of CO2 is kept at a minimum of 0.04%. That's what the carbon dioxide concentration in the air must be. And the only reason why it's kept at that percentage is because of the plants that utilize CO2 for photosynthesis. So that's the first raw material, right? And then we're going to look at what's the next raw material for photosynthesis. We're going to say it is actually light from the sun, right? So you can see the light from the sun together with CO2 from the air, and then you must get your water from the soil, which is H2O in symbolic form. So H2O is your water. Do you remember how water gets into the plant? Not just by diffusion, no, but it gets into the root, into the plants via the roots. So the roots actually absorb water, and roots do not have a cuticle. Do you remember in your first lesson you've done the structure of the chloroplast, and you've done the structure of the leaf, and you know on the leaf there's a cuticle that prevents water loss. Now there cannot be a cuticle around the root because the roots need to absorb water because we need it for photosynthesis. And then how does water move up the plant? Via your xylem vessels and your xylem tracheids. So all the plant tissues you learned in grade 10 is now going to come into effect in grade 11. You need to apply that knowledge to photosynthesis. So learners, let us look at this now again. Photosynthesis can take place at different speeds or at different rates. Depending on the concentration of the raw materials, photosynthesis will take place either slowly or fast. So it depends now how much of CO2 is in the air. How much of light is there? For example, for the past 2 days, you know it's during the day, but the light intensity was very low. We didn't see much of the sun. So automatically photosynthetic rate will drop. Right, so you're looking at CO2, you're looking at light, and then you're looking at water. We must be grateful that we had enough rainfall and that the grounds are now soaked with water, and it's ready for the roots of the plants to absorb that water for photosynthesis. Unlike in December, where it was very dry, there were no rains, and the farmers were very worried that all the mealy crops will die because there was not enough soil, not enough water in the soil for the mealy plants to take up water for them to photosynthesize and make your mealies. Yes, so photosynthesis, learners, remember, it's the only process that is able to make food for every human being, whether it's a nuts, it's proteins, it's your vegetables, your fruits, all of it is because of photosynthesis in the plants. Using what materials again? CO2, light, and water. Remember, this is very important to have that basic knowledge about photosynthesis. Okay, so now we're going to look at it, and remember, we're going to know what is the factory in the plant by which these raw materials are going to be used. It is called the chloroplast, right? It's called the chloroplast. Do you remember that? Now let us look at these factors. The factors that effect means what effect does this factor have on photosynthesis? So the factors that affect it, how slowly or quickly, how it takes place, it's going to be the light intensity. Light intensity means how bright is the light or how dull is the light. If it's cloudy, it means the light intensity is dropping or decreasing. If it's a very bright day without any clouds, it means the light intensity is quite high. Right, they're looking at the carbon dioxide concentration. Remember, we cannot keep a lot of carbon dioxide in the air because it causes global warming. And the ice glaciers in the Arctic regions are going to melt, and it's going to increase our temperatures. So we must always keep CO2 at its minimum of what? 0.04%. And the only process that can keep that CO2 minimum is photosynthesis. But what is pushing our CO2 levels higher and higher is the veld fires. Veld fires are actually a hazard to our environment, our ecosystem, and to the earth itself. So we must stop unnecessary veld fires. They must be put off because they increase the temperature of the earth, they are contributing to carbon dioxide increase, and they're causing global warming. So photosynthesis ensures that the CO2 level, the best level must be 0.04. And then also, children, the temperature of the air has to be correct for photosynthesis. So if you go to temperatures like 40 degrees or 35 degrees, it's very, very hot outside. This can be very harmful to the plants because they're going to be losing more water by transpiration than what they're going to absorb from the roots. So remember, when we're looking at photosynthesis, you don't just read and sort of, you understand the process as a whole. That photosynthesis means a lot to me. It means the plants are using photosynthesis to make food, and plus they release what? Oxygen. The most important gas on earth that sustains life is oxygen. The most important thing on life is food, and that two products, glucose and oxygen. Your glucose will form starch, it will form your beans, it will form your proteins, any food, right? It's coming because of photosynthesis. And that food we need it to survive, and then during photosynthesis we also release oxygen. But for photosynthesis to take place, we don't just need carbon dioxide, we don't just need enough sunlight, the temperature also has to be correct. It can't be too cold where the plants can get frosted and they die. It can't be too hot because then the water evaporation is going to be higher. So it must be at the best or the most suitable temperature. And I want you to learn a word, children, that the word for the most suitable is called optimum. We're going to come across that word just now. So let us just summarize the slide again. Photosynthesis can take place at different rates. That means speeds. Depending on the concentration of raw materials, photosynthesis can take place slower or faster. The factors that affect the rate, how slowly or how fast photosynthesis can occur, is because of the light intensity, the concentration of carbon dioxide, and the temperature. So you know this slide. Right. So now we're going to look at the influence of light intensity on photosynthesis. Now you're not going to just learn words, you're going to look at the graph. And learners, one important thing that you need to know or analyze from a graph is what is my x-axis and what is my y-axis. Remember, a graph line has a vertical line. We call that the y-axis. And your horizontal line, we call that the x-axis. Remember, the y-axis, there has to be a label. And that label line is always known as a dependent variable. A dependent variable. You're going to learn more about this in grade 12 when you're going to be doing investigations. That the y-axis is a dependent variable. Whatever is written on that y-axis, the vertical line, it means that that factor will depend on whatever is on the x-axis. Right, so let's see what's on the y-axis. Rate of photosynthesis means that rate of photosynthesis will depend on what is on the x-axis. What is written here? Intensity of light, or light intensity. That is your x-axis. And your x-axis is called your independent variable. You see, it's independent. Why? Because you as an investigator, you as a scientist, will manipulate this factor. You are able to now say, let me control how much of light I'm going to give to a plant, and let me see now if the rate of photosynthesis will increase or decrease. So you see, the rate of photosynthesis depends on the amount of light intensity. So you can't control how much of photosynthesis can take place, but you can control in an experiment how much of light you're going to give to a plant. All right, you can take a lamp, and then you can take a pot plant, a plant inside a pot. You can bring the plant closer to the lamp. It means it's going to get more light. You can take the plant further away from the lamp. It means the light intensity is going to decrease. So you can control the light intensity. That's why it's an independent variable. And what you can control, what you can manipulate, is always on your x-axis, and that factor that you're manipulating, that you're controlling, is called independent variable. Right? And the one that you're going to measure, you want to now measure the rate of photosynthesis. What you're going to measure, you don't know, it's going to change, it's not predictable. Then that rate or that factor that you have no control over, it's called the dependent variable, because the factor that you have no control over is depending on the factor that you are able to control. So here, let us analyze this graph now. This is a line graph. It's not a bar graph. It's not a histogram. It's not a pie chart. So this is a line graph. Right, learners? So here's the rate of photosynthesis as my dependent variable. And I can see at 1, my rate was very low. Can you see? As my light intensity increased to 2, even my rate of photosynthesis also increased. As my light intensity increased to 3, my rate of photosynthesis also increased. But then, as I increase the light intensity further and further, I see that my rate of photosynthesis stayed constant. So it increased from 1 to 2 to 3, but between 3 and 4, my line graph remained stable. It reached a point of equilibrium. So what does it mean? Which one was the most suitable light intensity? The light intensity at 3. Can you see? At 3, that was the most suitable light intensity, because at that point, that was the highest rate of photosynthesis. But when the light intensity was low, like here, the rate of photosynthesis was less. Okay, and then when it was at 4, it means it never increased or never decreased, but stayed at the same constant level. So how will we analyze this graph? We would say at low light intensities, the rate of photosynthesis is low. Now, learners, what I want you to see is that when you are analyzing a graph and you're describing the graph, the trend, you must always ensure there is a relationship between your two factors. Your factors are what? Your independent variable and your dependent variable. So what are my two factors that I'm investigating here? The rate of photosynthesis and the light against the light intensity. So that's my two variables or my two factors. When I describe the rate of photosynthesis in relationship to light intensity, I must describe it in the form of a relationship, like saying, as the light intensity increases, the rate of photosynthesis increases to a certain optimum. It doesn't just continue to increase. In other words, the graph line doesn't go like this. See, it's going to the highest point and then it is curving off. So at low light intensities, the rate of photosynthesis is low. As light intensity increases, the rate of photosynthesis also increases. So can you see there's a relationship between the two variables? As light intensity increases, the rate of the photosynthesis also increases. This happens to a certain point known as the optimum light intensity. Now they can ask you in the exam, learners, what is the optimum light intensity in this graph? So you're going to take your ruler and you're going to see what's the highest point. The highest point is not there, it looks like it's there. And obviously the points that you need to record will be numbered. Or sometimes it can be in between. But here you can see that this looks like the highest point, and that's the highest light intensity, and that's the rate of photosynthesis, which is at its highest, and that is at 3. And between 3 and 4, it remains constant because 3 was the optimum light intensity. Okay, so let's go on. Now we're still looking at light intensity. So here's the same graph line, light intensity or intensity of light, rate of photosynthesis. Again, the line graph is calibrated into 1, 2, 3, 4. And let's see what it reads here. When the light intensity is at its optimum now, we're not saying as it increases, when it's at the most suitable, that is 3, photosynthesis will occur most rapidly. So once the light intensity reaches the best level, that's when photosynthesis will be at its highest. So when light intensity is at its lowest, photosynthesis will be at its lowest. But now you can give a plant a lot more light, but it will be only at a certain point that it will take in that amount of light. Anything extra, the photosynthesis will not increase because it can only take in that best level. It's like, for example, you want to eat food, okay? And you see like cakes and sweets and cold drinks and chops and sausages and mealies and pap and bread and rolls and potato salad and coleslaw, all that at a braai. There's lots of food. You want to eat food for you to function. But can you eat all of that food, everything? No, because your stomach can only take a certain amount, optimum amount. It can't take any more than that. So therefore, even the plant, the chloroplast needs only a certain amount, the highest light intensity at a certain amount. Anything more than that, it will not increase the rate of photosynthesis. It will just stop there at its maximum, which we call optimum. So when light intensity is at its optimum, the photosynthesis will occur most rapidly. If the light intensity increases past the optimum, as I was saying, the rate of photosynthesis will remain constant. So when the light intensity is low, photosynthesis is low. As it increases, it also increases. But when the light intensity is at its optimum, then photosynthesis will be at its highest, and thereafter it will remain constant. And you can see the graph line is now leveling off. So the optimum light intensity is not 4, because between 3 and 4 it's constant. The optimum light intensity, the highest, will be at 3, because you can see that's the highest point. You can see there. Okay, here's the highest point. Right. Let's go to the next slide now. Now the influence of, now we spoke about light intensity. What we said were the factors? It was light, it was water, light, water, and it was also your carbon dioxide. So now we finished with light intensity. Let's look at the next factor. What influence will carbon dioxide have on photosynthetic rate? So here you can see now, I'm still having rate of photosynthesis as my independent variable. But now I want to test the effect of carbon dioxide. So I'm going to control how much of carbon dioxide I'm going to give to the plant. So concentration of CO2 at 1, 2, 3, 4. You can see that at 1 the carbon dioxide level is low, so photosynthetic rate is low. As we increase it to 2, okay, it can still increase. We go to 3, it seems like at 0.3 the CO2 level is at its optimum. That's the best amount of CO2, and then photosynthesis at its highest at 0.3. And we call that point what? The optimum level of CO2 for which maximum photosynthesis can occur. So at a low carbon dioxide concentration like here, the rate of photosynthesis is low at 1. As the carbon dioxide level increases, you can see as it's increasing, the rate of photosynthesis also increases. Can you see how you must have a relationship between the factor and the rate of photosynthesis? We want to know the effect of the factor that we're controlling, independent variable. What influence does it have on the factor we want to measure, the dependent variable? So the rate of photosynthesis depends on how much of CO2 is available for the plant. So as the CO2 concentration increases, the rate of photosynthesis also increases. This will happen up to its highest level, which we also call the optimum level. Okay, do you all understand that, learners? Right. Now let's go to the next factor. Here now we're still looking at carbon dioxide, but now we're seeing when the optimum amount of carbon dioxide is present, that means when the highest amount of CO2 is present, what happens now? The photosynthesis will occur most rapidly. And look at the wording. It doesn't just say photosynthesis will increase. It will say photosynthesis will occur most. That means the highest amount of increase. So it will occur most rapidly, or you can say the highest rate of photosynthesis will occur when the rate of CO2 increases to its optimum level. The rate of photosynthesis will also increase to its maximum level. Okay. So if the carbon dioxide is higher than the optimum, like if it's, we're continuously giving it CO2 at 4, maybe at 5, we're carrying on giving it, but the graph line just levels off. It doesn't go more than that. It doesn't peak like that. It doesn't. So what is the best level? It's going to be the highest point here, which is going to be 3. Okay. So if the carbon dioxide concentration is higher than the optimum, like higher, then the photosynthesis level will remain constant. This is because the light independent phase, the dark phase, or the Kelvin cycle, uses the maximum amount of CO2. So learners, just to recap again, do you remember when your teacher first spoke to you about photosynthesis and you did an equation? What are the raw materials for photosynthesis and what are the products? And then, which of the raw materials are used in 2 phases? Do you remember the phases of photosynthesis, learners? Do you remember? Okay, it is what? It is the light phase. And why is it called light phase? Because it depends on light. It needs light, okay? Although the light is there, it must need the light. And the other phase is called the dark phase. And please note, learners, that the other phase of photosynthesis is called the dark phase, not because it must happen in the dark. The dark phase will still occur during the day, but the reason why it's called the dark phase or Calvin cycle is because it does not need light. It can occur in light, but that phase does not require the light. It will require another raw material. Okay, so we're going to do that equation just now. So let us see. This is because the light independent phase. So what's the light independent phase? The phase that does not depend on light. It is called the dark phase or Calvin cycle. Which raw material will that need? It will need CO2. Okay. So now, learners, let's look at it here. You're going to see, okay, let's look at the next slide. Right here, I'm going to just draw for you that in the light phase, all right, we have now, remember your chloroplast, which was disc-shaped. You remember it had these sac-like structures called lamella, which were stacked on top of each other, like this. And you remember that these lamella were membranous sacs, right? They were membranous sacs like this. And what were they filled with? They were filled with a green pigment called, good, chlorophyll. Now, what is the main purpose of chlorophyll? Chlorophyll is the one that absorbs your red and green light. Remember, light is white, but there are different wavelengths of light. And you find that the red and the green wavelengths are used to the optimum level. So this chlorophyll actually absorbs your red and your green light, right? From your 7 wavelengths of light, the chlorophyll absorbs the light, and the light is going to energize the whole process of photosynthesis. So that light energy of the sun is going to start activating the whole process of photosynthesis. So you're going to see now that this is the light phase, and in the light phase we need chlorophyll, and it's the chlorophyll that needs light. It absorbs light. That's why it's called the light phase. Now, in the light phase, what was produced? Oxygen was made, learners. Do you remember that when the light strikes the chlorophyll, it energizes the electrons, and then you're going to have your glucose being formed, right? And you're going to have your oxygen, your water is going to be used, and your water is going to be used, and the oxygen is going to be released in the light phase. Remember, in the light phase, what is released? Oxygen. Let us do the whole equation here now. Light plus water from the soil plus CO2 from the air is going to give us C6H12O6 plus oxygen. These are the products, and these are the raw materials. So, which raw material is going to be used in the light phase of photosynthesis? It's going to be light and water. Right. So now, which one is going to be given off as a product in the light phase? It's going to be oxygen. Which means now in the dark phase, which of the raw materials we're going to be using? We're going to be using CO2 in the dark phase. See, we're not using light. Light was used in the light phase. That's why it's called light phase. But the dark phase can occur immediately after the light phase. When the products of the light phase is available, then immediately it goes into the dark phase. But it's called the dark phase, and they'll ask you this in the multiple choice. It's called the dark phase because it does not require light, not because it occurs only at night. Remember that. So this dark phase requires CO2, and what we're going to get eventually is going to be your carbohydrates and your proteins and your oils, which are your fats. and all your castor oils and your sunflower oil that comes in the sunflower seed, and your nuts, which are proteins, and your starch, which is your wheat and your rice. All those foods are carbohydrates, and they are coming from photosynthesis because of the CO2 that's absorbed in the air. That CO2, the C's are going to form your C's in glucose, and then your H is going to be the H is coming from the hydrogen in water. Sorry, learners. And you find that also the O is going to be forming part of your glucose or your carbohydrates, right? And that oxygen as well over here from the water, it's going to be released during the light phase. Okay, now that we've seen this whole thing, let us look at the slide one more time, right? And we're going to see now, okay, we're going to see the effect of temperature. Let's see. Temperature influences the rate of photosynthesis. Remember, we discussed light intensity. As light intensity increases, photosynthetic rate increases. As light intensity increases to its optimum, photosynthesis will be at its highest level. If there's more increase in light intensity, photosynthesis will remain constant. CO2, the higher the CO2 level, the higher the rate of photosynthesis. The highest amount of CO2 that's used will be the optimum rate of photosynthesis. Anything higher than the optimum CO2 levels, photosynthesis levels will remain the same. Low levels of CO2, photosynthetic rate is low. Low levels of light intensity, photosynthetic rate is low. Now let's look at temperature. Now this graph gives you a perfect example of a dome or like a hill. So it goes from 1, 2, 3, and 4. Now you can see that the entire graph is not just going constant, it's going up and then comes right down to 0. So learners, any point here on the graph line, it is 0. Okay, so here's your rate of photosynthesis and here's your temperature line, okay? What do they say? Temperature influences the rate of photosynthesis. And learners, you would notice when it's very cold, you will not be as active as when it's very hot. But if it's too hot, on the other hand, you become lethargic, you become tired. You like it to be hot, but if it's too hot, you can't function. If it's too cold, you can't function. But if the temperature is just suitable, like 25 degrees, then you find you can do a lot of work without getting tired. The same thing occurs with a plant. In a plant now, the rate of photosynthesis is going to increase as the temperature increases. Look, the rate of photosynthesis is increasing. As the temperature increases, the rate of photosynthesis is increasing to its optimum level here, which is actually 3. Now, temperatures higher than that, what is going to happen? You find the rate of photosynthesis after the optimum, the rate of photosynthesis is going to drop. It's going to decrease. Learners, do you remember the reason why, what happens when plants or enzymes are exposed to very high temperatures? Think about it now. Okay, learners, do you remember what happens to plants when they're exposed to very high temperatures? Yes, the enzymes are going to denature. Can you see that word? So let us look at it. When temperature is at its optimum, that is at level 3, the rate of photosynthesis will reach a maximum. If the temperature is higher than the optimum or maximum, then the photosynthesis will decrease in rate. See, when you're giving more higher temperature, instead of the photosynthesis increasing, it's going to now start decreasing. This is because the enzymes used in the process will denature at high temperatures and will no longer function. Now, learners, what is an enzyme? It is a substance that will start off a reaction. It's like a key. And an enzyme works on a lock and key basis, as what you learned in grade 10. So let's say now you have a key to open the door, right? You can open the door and close the door with the same key as many times as you want. But now, if you take the key and you put it into a freezer and you use it, it'll still work. But you'll struggle because the key is too cold. If you take the very same key and you put it into a fire, what will happen to that key if it's a metal? It's going to melt. And when you take that melted key and cool it off, what's going to happen to that key? It's going to change its whole shape because you exposed it to very high temperatures. When the shape changes, we say that the enzyme will denature. So when enzymes are given too high temperatures, they change the shape so they can no longer function. And then photosynthesis will not occur because now the enzymes for photosynthesis have denatured. So that is what's happening between points 3 and 4. The photosynthetic enzymes denatured due to very high temperatures, and therefore photosynthesis rate will drop. Okay. Now let's go to some activities which are a little bit more difficult. Okay, learners, now here we're going to look at the mean mass of lettuce plants after 21 days. Now we're going to look at how much mass is there in this lettuce. Remember, when plants photosynthesize, they must make food. So the amount of food made in the plant is going to be the mass. So how many beans did you have? How much of starch is there in the meal, in the rice? How much of starch is there in the potato? How much of glucose is there in the lettuce? It's because of the food, the glucose made in the dark phase of photosynthesis. Remember, you did the dark phase. And we said in the dark phase, it is glucose that is made. It's carbohydrates that's made. So now, you want to measure how much of carbohydrates have been made after the light phase and the dark phase took place. Complete photosynthesis, how much of food was made. So now you're going to look at the mass. The mass goes from 0 to 100 to 200 to 300 to 400. Now this graph line, learners, is showing you 3 different factors on one graph line and how these 3 factors affect the mass. So let us see. Here they're talking about light intensity. As the light intensity increases, how it influences the mass of the lettuce. But it's also telling you that on these graph lines, we're also giving it different amounts of CO2 at different temperatures. Okay, you can see the light intensity, they're showing you 2, 4, 6, 8, 10. That's the different strengths of light intensity. And here they're showing you at different light intensities how much of CO2 we are giving at different levels and at what temperature. So let us look at this lowest line. This is 0.03% CO2. That's not the optimum. What did we say the optimum CO2 level was? 0.04%. So 0.03 is a little bit less than 0.04, which means that the level of CO2 there is quite low. So photosynthetic rate is low. So less carbohydrates are made in the lettuce. Therefore, the mass is low. Also, the temperature there is 15 degrees. So when you gave it 0.03% CO2, you kept that plant at 15 degrees Celsius. And the best level for photosynthesis is around 25. Now 15, it means the enzymes cannot function. It's like too cold. So therefore, the photosynthetic rate is very, very low. It's almost like less than 50. See, if that's 50, it is far less than 50. Far less than 50 milligrams or 50 grams of of the mass of glucose was made in the lettuce, right? Can you see? That's even less than half, less than 50. Because the CO2 level is low and the temperature is low. Therefore, the rate of photosynthesis is low. Therefore, there's less mass of carbohydrates in the lettuce. Let's look at the next graph line. Here now, we're giving it again 0.03% CO2, but we're keeping it at 25 degrees. Right, so we're increasing the light intensity, but again, the CO2 level is not the suitable one. It's 0.03. So the photosynthetic rate did increase because you increase the temperature. You kept the CO2 level same, you increase the temperature, you kept the light intensity the same, but because it wasn't the optimum 0.04 CO2, the rate of photosynthesis increased a little bit, and the mass of in the lettuce also increased compared to at 15 degrees, but not too much of mass. Okay. Then we're going to now the next graph line, which is the dark line here. Here it is actually 4%, right? 4%, and that is going to be 0.04, and that is the optimum level. It's CO2 at 0.04. 4 over 100 is equal to 0.04 in decimals. And we know that that is the most suitable level of CO2. So when you're giving this plant 0.04% CO2, but you kept it at only 15 degrees, now that's too low. So you're giving it the right CO2, you're giving it the right light intensity, but now the temperature is low. So although the optimum CO2 level is increased and the light intensity, but because the temperature is low, the enzymes can't function to the optimum. So the photosynthesis rate did increase, but not to the highest amount, but it did increase compared to when it was at 0.03 and it was at 25 degrees. So here the percentage of CO2 is correct, but the temperature is incorrect. So let us look at the last line now, this last dotted line. Here it is 0.04% CO2, which is 4%. That's perfect. You're giving it enough light intensity, and what's the most suitable light intensity? The most suitable one, the highest mass of lettuce is going to be here. And you're going to take your ruler and you're going to read it off like that. It's almost like 397 grams. Can you see? It's almost 397 grams of mass of lettuce. That's higher than this one, which is about 302. That's 397. So that's the highest point of mass, highest rate of photosynthesis, because the CO2 level was correct, the temperature was correct, it was the optimum light intensity of 8 units. That means the optimum light intensity is going to be 8 and not here. Okay, it's not going to be here by 6.5. It's going to be, the highest is going to be at 8, because that's the highest mass of lettuce. And the temperature is correct, 25 degrees, not too cold, not too hot, correct CO2 levels. So therefore, this graph line here is showing you the optimum level of photosynthesis, giving you the optimum amount of mass in grams in the lettuce, the highest amount of carbohydrates produced in the lettuce after the light phase and the dark phase. Okay, so let us see. Scientists set up an apparatus to investigate the effect of temperature, light intensity, and carbon dioxide concentration on plant growth. Now, learners, always, we first analyze this graph because we've just done those graphs and we took that graph and we applied it to this graph. Now, when you're reading it, it makes a lot of sense that the scientists wanted to investigate the effect of temperature, there's it in degrees Celsius, light intensity here in units, CO2 levels in percentage, right, on plant growth. Using this apparatus, they could control each factor. The scientists set different temperatures, and what were they? What were the 3 temperatures that they set it at? Actually, it's only 2 temperatures. It was 15 degrees and 25 degrees. Okay. CO2 concentration, there was only 2, 0,03 and 0,04, okay, or 3% and 4%. And light intensity for 4 different groups of lettuce plants. What are the 4? It was A, B, C, and D. You could also say that that was A, B, C, and D. They didn't give you the label lines, so you can assume that those are the 4 graphs, A, B, C, D. Okay. You always start off from the bottom and then you go to the top. The average mass of lettuce plants serves as an indication on the rate of photosynthesis. So this was the highest rate of mass in the lettuce. So that's the highest rate of photosynthesis. Let us look at the same graph, and now let's look at the next set of questions. I'm going to give you 2 minutes to take some time to answer the question before we look at the answers. What is the influence of light intensity on the average mass of the lettuce plants? 3 marks. Please try to answer this question, children. What is the influence of light intensity on the average mass of the lettuce plants? Okay, welcome back again, learners. Did you get the answer right? What is the average influence of the light intensity? And you must say it like this, that the rate of photosynthesis increases. Did you have that? The rate of photosynthesis increases if the light intensity increases. Therefore, the average mass of the lettuce plants will increase. Okay, you could have also, if anyone answered it in a different way, you could have also said, as the light intensity of a plant, as the light intensity will increase, so does the rate of photosynthesis, which will also mean that the mass in the lettuce plants will also increase to an optimum. Okay, let's go to the next one. Name 2 limiting factors that influence the rate of photosynthesis as the light intensity increases. Now, children, this is a very important point, because we are not only looking at one factor that controls photosynthesis, we are looking at 3. Light intensity, CO2, and temperature. So, what are the 2 factors that will limit photosynthesis, although you may give the plant light? The plant does not only need light, it also needs CO2 and temperature. So, the 2 factors that can limit it is CO2 and temperature, because you're giving the plant light, but if it's not the right temperature, if it's not the correct CO2, then you're going to affect photosynthesis. So the answer there is carbon dioxide and temperature. You need all 3 factors for the optimum rate of photosynthesis. If you only give it carbon dioxide, then what will be the 2 limiting factors? Then it's going to be light intensity and temperature. If you only expose the plant to a high temperature without light, without CO2, then photosynthesis will not take place, because you're only giving it temperature, but you're not giving it CO2 and light. So photosynthesis will not occur, because it needs all 3, and all 3 needs to be at the best, the most suitable level, not too low and not too high. If you're giving the plant only, for example, now carbon dioxide and no light and no temperature, and you keep it very cold, there's no temperature, it's just like very low, and you're only giving it carbon dioxide, right? What's going to happen? Then again, photosynthesis will drop, because all 3 factors are not present in the optimum levels. Right, so you can see there's 2 marks for there. Name 2 limiting factors. It's carbon dioxide and temperature. If they ask you, learners, to explain the 2 limiting factors affecting the rate of photosynthesis as light intensity increases, when they ask you to explain, you cannot just list a factor. then you got to give a reason why CO2 and temperature are limiting factors. Then you would say, a plant needs the optimum amount of light intensity for optimum photosynthesis to produce an optimum mass of carbohydrates in the lettuce. Therefore, if there is no optimum CO2 or no optimum temperature, this will decrease and affect the rate of photosynthesis and will also affect the mass of the lettuce plants. There you've explained. You've explained the factor and what it has on the effect of photosynthesis and on the mass, because this whole graph is dealing with photosynthesis rate and as well as the mass. So you got to link all the factors to get full marks. All right. Now, how were the scientists able to increase the rate of photosynthesis to the maximum level? Okay, learners, I'm giving you a few minutes to answer this question. How were the scientists able to increase the rate of photosynthesis to the highest level? Look at all these graphs here and see the highest level and decide now what will be your best answer for 3 full marks. I'm giving you just 2 minutes to answer, and then we're going to check your answer against the correct one. Right, welcome back, learners. I hope you've answered that question, and let us see your answers again now. Right, how were the scientists able to increase the rate of photosynthesis to the maximum? What is another name for maximum when we're talking about photosynthesis, or the most suitable? We use the word optimum. Don't forget, learners, we must use the word optimum, okay? Optimum instead of maximum. Because maximum means like full, very, very full, the highest. But we know we cannot give it the highest. We need to give it the most suitable out of the highest. That's why we use the word optimum. So if there's a question that says, why can't you say the maximum amount of light? Because photosynthesis can only use light to a certain degree, not to the highest amount, but to a certain point. That's why we use the word optimum. So let us look at it now. The scientists, what did they do? They increased the CO2 level to an optimum level of 4%. And you can say the scientists increased the CO2 level from 3% to 4%, which is the most suitable or optimum level of photosynthesis to yield the highest amount of mass. You heard what I said, learners? You can say they increased the CO2 level from 3% to 4%, which is the optimum percentage of CO2, thereby increasing photosynthesis to its optimum and also increasing the mass of the lettuce plants to its optimum. But besides that, what else did the scientists do to ensure that the rate of photosynthesis was at its maximum, the highest? The scientists not only increased CO2, but they also increased the temperature from 15 degrees Celsius. What is this unit? It's degrees Celsius. Whenever you talk of temperature, you must use the correct unit. When you talk of mass, you must use the correct unit, which is grams or kilograms. That's very important. When you're looking at light intensity, you talk about arbitrary units. Arbitrary units like 1, 2, 3, 4 arbitrary units. That is light intensity. Okay. So let's look at it, children, now. They increase the temperature from 15 degrees to 25 degrees Celsius. And therefore, this 25 degrees is the optimum temperature, increasing the rate of photosynthesis to its optimum and also increasing the mass of the lettuce plants to its optimum. But what else did they do? They didn't only increase the CO2 level from 3 to 4, temperature from 15 to 25. What else did they do? They also increased the light intensity from 2 to 8. Can you see? 8 was the highest level of light intensity. And they also increased the light intensity from 2 arbitrary units to 8 arbitrary units, which is the optimum level of light intensity, therefore increasing the CO2 level, increasing light intensity, increasing the rate of photosynthesis, and increasing the mass of the lettuce plants. Okay. We now look at the very same graph, learners, and this is an application question for 1 mark, but you need to read it and see what answer you get. I'm giving you another minute or 2 to answer this question. Remember, you must analyze your graph, and in life science, the way your answer is formatted is very, very important. It's not a general answer. Your answer must be scientific. It must show more photosynthesis, highest amount, lowest amount, increase or decrease. When you talk of temperature, you use the units. You talk of light intensity, you use the units. If you miss any of those, you may think you have the answer right, but it will be wrong. So please, learners, before you answer this question, think of exactly what must I put into my answer to get all the marks that are on the question paper. I'm giving you 2 minutes. Right, welcome back, learners. Did you answer the question on what will happen to the rate? And remember, now we are looking at 2 things here. We're looking at the rate of photosynthesis, and we're looking at the temperature being raised. And not just raised, but raised beyond 35 degrees. Now, remember, what was the lowest temperature? 15. What was the optimum? 25. Now they're saying, we're going to give this lettuce plant and keep it at a temperature of 35. Now, what's going to happen to that rate of photosynthesis? It's going to decrease. It's not going to increase, it's going to decrease. Why will the rate of photosynthesis decrease if the temperature is going beyond 35 degrees Celsius? Good. The enzymes are going to denature at high temperatures. Therefore, the enzymes controlling the dark phase of photosynthesis will denature. They will no longer be able to manufacture carbohydrates, and the mass of carbohydrates in lettuce will decrease because the rate of photosynthesis will also decrease due to the denaturing of the enzymes. So you see, that's what you must have in your answer. So let us see how was the answer. The rate of photosynthesis will decrease drastically. You see the word drastic. They never just say the photosynthesis will drop. They say the rate of photosynthesis will drop drastically. What could be another term for drastic? You can say the rate of photosynthesis will decrease sharply. Sharp decrease, a very fast decline. Can you see that? Not just decrease like slowly. So the rate of photosynthesis will decrease drastically or sharply. Now, that is because it's just 1 mark. They just want to know what would happen to the rate. They didn't ask for explaining. They said, what would happen to the rate of photosynthesis if the temperature is raised beyond 35 degrees Celsius? Let your mark allocation also guide you to the answer. It's only 1 mark. They want to know what would happen to the rate if the rate of photosynthesis will decrease. You won't get a mark. But if you say it will decrease rapidly, that means it will decrease but sharply or rapidly, very fast. It's going to have a sharp decrease. Then you get, because now you are describing the decrease in an appropriate manner, because it's going from 25. I mean, if you are sitting at a normal temperature of 25 degrees and now your temperature increases to 35 degrees, you're going to start sweating. At 25, you don't sweat, but at 35, you start sweating. and the heat becomes intolerable. So same thing for the plants. If it's 35 degrees, it becomes too hot. Now they're losing water. Instead of keeping water for photosynthesis, they're losing water. And therefore, because the water levels are going to drop in the chloroplast, less water will be absorbed, more water will be lost. Photosynthesis is going to decrease, okay? Now, learners, we need to give a reason. You see now, give a reason is the same as saying explain your answer for why photosynthesis rate will decrease sharply or drastically if it's exposed to 35 degrees. So give a reason for your answer in the previous question. And you can see that high temperatures denatures enzymes. They become inactive. Okay. What happens here? The high temperatures will denate. You get a mark for saying, because you have to relate to the previous one. You can say high temperatures of 35 degrees Celsius is above the optimum of, you can say, high temperature of 35 degrees Celsius is above the optimum temperature of 25. Remember, so the high temperature of 35 degrees Celsius is higher than the optimum of 25 degrees. Therefore, the enzymes will denature, they become inactive, and this will drop the rate of photosynthesis. Okay. Now, this is another activity dealing with another experiment which you do in photosynthesis. Can you remember this diagram? Can you remember your teacher doing this experiment even as a demonstration in the classroom? Here is a light or a lamp, and what is it giving off? It's giving off the light intensity. But remember, you can also put a thermometer here, because this is, what is this, learners? Look at the labels. This is a beaker containing water. This is a test tube. So here's your test tube, and you're collecting a gas in this test tube over here. There's your beaker and water, and you're going to put a water plant. Why do you need a water plant? Because it does not have a cuticle. So what plant are you going to be using in this experiment? You cannot go and break a plant from the garden and use it for this experiment because it's got a cuticle, and then water won't go in through the leaves, and if there's no water, there'll be no photosynthesis. So you've got to get a water plant where there's no cuticle for the CO2 to diffuse through, for light to go through easily, and for water to be absorbed through the leaves to go to the chlorophyll in the chloroplast. So here is now an elodia. This plant that's photosynthesizing, can you see it's giving off tiny little bubbles? Can you see all the bubbles that are rising? What gas does photosynthesis release? Yes, the most sustainable gas on earth. It sustains life. Without 4 minutes, we would die. What is the name of the gas? Oxygen. So these are oxygen bubbles that are rising from this plant. We can have this plant. It's also called an elodia. Okay? We use an elodia. It's a water plant. You can get it from a pet shop where they have a little fish aquariums. That's where they put the water plants. So you can go there. You can the shopkeeper there will actually give you a piece of the plant and maybe charge you a small amount. You take it, you put it into a beaker of water, then you take a glass funnel. It must be glass because you want light to go through. You do not use a plastic funnel. So this is an inverted funnel, but it must be a glass funnel. If you use a plastic funnel and that plastic is not transparent, light will be blocked. So you can give the plant a warm temperature, you can put a lamp there, but the light will not reach the plant because you are using a plastic funnel. So the funnel has to be a thin glass funnel, which you get in the laboratory. This funnel at the top is opened. So you simply put a little bit of Vaseline on the sides because you don't want the gas that's collected to be actually escaping. So you put a little Vaseline on the sides of that glass funnel and you invert a test tube which is empty over that glass funnel. You keep some water that is submerged inside this beaker. This water is here, all water here. The water is filling up in the beaker and the water fills up till that stage there. Right, that's where the water is. And then automatically bubbles will start moving up. And as the bubbles move up, it will push the water down. It's because gas displaces water. So as the gas bubbles rises, it's going to accumulate on the top here, and it's going to push the water down. When all this water goes down, you've got to quickly take the test tube, seal it with your thumb because you don't want the gas that's collected to escape. Seal it with your thumb, and you want to test now. Is the gas oxygen? Leonard, I'm giving you 2 minutes to think about how would you test for oxygen gas. Are you ready, learners? Can you tell me what in grade, I think you've done it in grade 9 when you did the testing for the different gases using magnesium ribbon, and you tested for sulfur, you tested for hydrogen with a popping sound. Your teacher must have also showed you how do we know that a gas that's released is oxygen, because can we see oxygen in the air? No. It's an invisible gas. So as the water bubbles were rising in the test tube, the water went down. So what pushed the water down? Not a ghost. The air bubbles of oxygen pushed the water down. So when you're removing the test tube, you've got to quickly seal it. And then we use what we call a glowing splinter. Good. We use a glowing splinter. You're going to take a glowing splinter. You can even take a little matchstick. You light it, then you quickly blow it off, and while the tip is still red hot, you put it into the test tube and you'll see it will burst into a flame. Even if you have an incense stick, while the tip is burning red and the smoke, you take the incense stick and you immerse it into the into the test tube. Deep down, you'd see the incense stick bursting into a flame. You need to know that that gas is oxygen. Oxygen supports burning. What's another name for burning? Combustion. Good. So let us read this experiment now, sons. The diagram below shows the apparatus used to investigate the rate of photosynthesis in a submerged plant. When you see the words to investigate the rate of photosynthesis, right, and its effect on the oxygen bubbles, you must know that is an aim, to investigate whether this and this has an effect on that and that. That becomes your aim, to investigate the rate of photosynthesis on the production of oxygen bubbles. Okay? Now the light intensity to which the plant is subjected was decreased at regular intervals by moving the light source away from the plant. The average number of bubbles per minute was counted and recorded for each distance. Now you have a table alongside your diagram. Here's the lamp, here's your apparatus, here's your Elogia plant. You're giving it light for photosynthesis. It's transparent for light to pass through. You're also going to put a little bit of bicarb inside this water, because bicarbonate of soda is a source of carbon dioxide. Okay, it's going to give you carbon dioxide. You're getting light. You can also put a thermometer into the water to keep it at 25 degrees Celsius. So you got your temperature being correct. You got your light intensity that you're going to change. And now what you want to measure is the production of oxygen. How light intensity affects the production of bubbles. Because remember, oxygen is produced in the light phase, which requires light, That's where oxygen as a product of photosynthesis is produced. Now let us look at this table of data. Here they are telling you that at the distance of lamp from the plant, when the lamp is 10 centimeter, that means very close. 10 centimeter is quite close to this apparatus. There were 60 bubbles of oxygen recorded. When it moved away, 20 centimeters, still 60. 30, still 60. So which means that the best level of photosynthesis for this plant to produce maximum oxygen bubbles would have been 30 centimeters. So if you move the plant at 30, that was the most suitable, okay? Because at 10, 20 or 30, the oxygen bubble remains the same, 60. That means the highest amount of oxygen, the highest rate of photosynthesis when it was brought very close. But even between 10 and 30, the oxygen bubbles remain the same. But the minute you took it further away, 40 centimeters away from the apparatus, 50, it was still 60, right? But when you moved 50 centimeters away, the number of bubbles dropped. 60 centimeters away, you can see it was 50 bubbles. When you moved further, 70, 45, 80, 40. When you moved almost 3 rulers away from the apparatus, you only got 35 bubbles. But when you moved at 100 centimeters, you only got 30 bubbles. So the highest rate of photosynthesis for the highest amount of oxygen bubbles was at 10 centimeters. That's the closest, okay? At 10 centimeter, that's the closest, you got the highest amount of bubbles. So can you see that even if you went further away, or even, and you can actually say 30 was the best, because even when you brought it closer and closer, the rate of bubbles remained the same. So when you moved at 30, that was the highest amount of bubbles. So at 30 centimeters, so as you took it from 100 centimeter to 90, then when you brought it at 30 centimeters, you've seen that was the highest amount. When you brought it to 20 and 10, it still remains 60. So the most suitable distance was at 30, because even if you brought it closer, it still remained at 60 bubbles per minute. Okay, learners, so here the average number of bubbles per minute was counted and recorded for each distance. So how was the rate of, now they're going to ask you a question. How was the rate of photosynthesis measured in this experiment? We never used the mass of letters, but what did we use? We used by counting the number of oxygen bubbles. That's how we measured the rate of photosynthesis using light intensity, by looking at the number of oxygen bubbles that we produced. And that is how it was measured, by counting the number of oxygen bubbles per minute, depending on the distance of the lamp from the apparatus. All right, so here's your experiment. Now here are some questions. Okay, what is the aim of the investigation? I told you, learners, an aim will always start off with to determine whatever or to investigate. Okay, so now I'm giving you 1 minute to answer this question. What is the aim of the investigation? Okay, learners, we come back. What will be the aim of this investigation? You're looking at bubbles, you're looking at light intensity, so what will be the aim? And then name the gas released by the plant. The aim is going to be to determine or to investigate the effect of light intensity on the rate of photosynthesis. Can you see that? To determine the effect of light intensity on the rate of, so what effect does light intensity have on the rate of photosynthesis? But how did we measure the rate? We measured the rate by counting the oxygen bubbles per minute. Okay, so name the gas released by the plant. What is the name of the gas released? It's going to be oxygen. Okay, so let's look at the next question. Describe a test to verify your answer in the previous question. So learners, what's the previous question? Name the gas released by the plant. We know plants can only release one gas, and that is oxygen due to photosynthesis. So this question is saying, describe a test to verify your answer. How are you going to indicate to somebody that that gas released is oxygen when you can't even see the gas? It's invisible. How are you going to show them? How are you going to show proof that that is oxygen? Because according to tests and investigation and laws and evidence, we know that anything that has oxygen, there will be a fire. If there's no, if something cannot burn, it means there's no oxygen. So when we are lighting fires, the fires that you are lighting on as a fell fire, it's using up valuable oxygen. What we're supposed to inhale and get better with, the fire is using up that oxygen and lighting wildly. So the best way to test to verify oxygen is by using a glowing splinter. Immerse a glowing splinter into a test tube in which the gas is collected. See, you got to describe. You don't just say a glowing splinter. Describe it. You immerse, that means you can put a glowing splinter. What's a glowing splinter? A stick that has a glowing end of the that is burning red at the end. So immerse a glowing splinter into the test tube in which the gas is collected. If the splinter bursts into a flame, it shows that oxygen is present. Can you see? It's a full sentence of how you're going to describe it. You don't just say, use a glowing splinter. You say, use a glowing splinter, put it into the test tube where the gas was collected, and if it bursts into a flame, it will indicate that the gas released was oxygen. Okay, now describe the relationship between the gas released and the light intensity. So the gas released and the light intensity. Now learners, think about it. How will you describe the relationship now? You're going to look at this table of data. You're going to look at the relationship between gas released and light intensity. That means if the plant or the apparatus or the lamp is closer to the plant, that means if this lamp is 10 centimeter away, a small, here's the apparatus, here's it, and it's 10 centimeters away, 60 bubbles. If I put it 20 centimeters, 60 bubbles, 30, 60, which means that when I put it at 100 centimeters, very, very far, almost 3.5 rulers away from the apparatus. There's the apparatus. If I put it 1, 2, 3.5 rulers away, I get less oxygen bubbles because they're talking about the gas released. So the further the light, the further the lamp from the apparatus, it means the light intensity decreases, then less oxygen bubbles produced. As the light intensity increases, when you bring the lamp closer, more oxygen bubbles were released. So let us see the answer. You see, it's a relationship. When the light intensity decreases, for example, further away from 30 centimeter, the number of bubbles released also decreases. So the further the lamp from the apparatus of the plant, light intensity decreases because now it's becoming dull, it's too far. Rate of photosynthesis decreases and the number of oxygen bubbles will also decrease. Okay. Name 2 other environmental factors except light intensity which you think could change the rate of bubble production. Remember, learners, what we said. You're just giving this plant light and obviously water. But what else you need for photosynthesis? You need CO2, but we know in this experiment here, CO2 is not needed for oxygen production. CO2 is only needed in the dark phase for the production of glucose. So in the light phase, it's where oxygen is produced. But I can give the plant a lot of light. Okay, there's no CO2, it's not a problem. I'll still produce my oxygen. I'm just not making carbohydrates. But what else does this plant need for the enzymes to function, to produce, for light phase, to produce the oxygen bubble? You need temperature, isn't it? So the two other environmental factors must be, the temperature must be correct, which is what? 25 degrees. Plus, eventually, for the entire photosynthesis to produce, you also need CO2. So name two other factors except light intensity which you think could change the rate of bubble production. Bubble production. It's going to be temperature and carbon dioxide concentration. Although temperature is the main one that affects the bubble production, because it is the temperature that's going to cause the enzymes that control photosynthesis to function well at a suitable temperature, optimum one at 25 degrees. So that's when maximum optimum photosynthesis, optimum number of oxygen bubbles, which was what? 60, right? And you can also say carbon dioxide because the full process of photosynthesis must occur. You need CO2 as well. Also, you need chlorophyll, isn't it? But they're saying environmental factors, so you can't say chlorophyll because chlorophyll is a structural factor that affects photosynthesis. It's not an environmental one. So because it's environmental, you need to say that it's temperature, and also you can say water. Remember, they said besides light intensity, we need light, yes. We need temperature, yes. And we also need water because it is the light energy that splits this water into hydrogen and oxygen. And that's the oxygen bubbles that we see being released. And this hydrogen is going to be carried in with the CO2 to form your carbohydrate, your C6H12O6. So this hydrogen released from water goes into the dark phase. Got nothing to do with oxygen bubbles. But this water, when it's split, it releases the oxygen bubbles. So what you need, what are the two factors you need for oxygen production is going to be not just carbon dioxide, but temperature and water. That is the light phase. It needs the correct temperature and water because water is split to give us oxygen. So learners, now you know the importance of rain. That without water in the soil, you won't get oxygen. And without light from the sun, the light of the sun, if there's no light of the sun, it can't split the water. If you can have sunlight, but no water, you can't get oxygen. So they are dependent upon, if there's light and if there's water, but if it's too cold, then again there's going to be less oxygen. So we need the correct temperature, the correct amount of water, and the correct amount of light intensity for maximum oxygen production. We need the correct temperature, we need the correct amount of water, we need the correct amount of CO2 for the maximum amount of glucose for mass production in a plant, to produce maximum fruit, to produce the best fruit, big apples and big pears and big potatoes. For maximum photosynthesis, all the levels of CO2 and water and light and temperature must be at its highest, okay? So let's go to the next one. So learners, this brings us to an end of the lesson for photosynthesis, or the factors that affect the rate of photosynthesis. Please, learners, analyze your graphs and always use these equations and know that the two phases is the light phase and the dark phase. What you need to know, and that's going to be tested in the June exam, right? You're going to write one paper, is what are the products in the light phase and what are the raw materials used in the light phase. So in the light phase, it's light plus water from the soil, right? And what it gives you is oxygen, because the light energy splits the water to give you oxygen. Then in the dark phase, it's dark not because it occurs at night. It's a dark phase because it does not require light. So what are the raw materials in the dark phase? It's CO2 plus the hydrogen that comes in from the light phase. the splitting of water. See, we release this oxygen into the air, but this hydrogen that's released from water joins with the carbon dioxide to give you what? Glucose. So you must put that in a full equation. It's going to be light plus water plus CO2 is going to give us glucose, which joins to form your starch and your proteins, to form all your foods and your oils, right? That is, your glucose is going to join with hydrogen, more hydrogens, and more to give you starches, and then it's going to join with nitrogen to give you proteins, and it's going to join with more oxygen and more hydrogens to give you fats. So that's going to give you food, which is glucose and oxygen. And you can see this one is made in the dark phase, and this one is made in the light phase. And these two are used in the light phase, and this one's used in the dark phase. So if you can put the entire equation together, you can understand that this whole process also needs the correct temperature, and the correct or optimum temperature for plants to photosynthesize is going to be 25 degrees Celsius. Now, learners, you're also going to learn about another process which is very similar, and we're going to do it in the coming weeks, and that is respiration. And that respiration and photosynthesis work hand in hand. The processes are very similar, and therefore you need to know all your facts of photosynthesis. That what is the factory? It's the chloroplast. What is the pigment? It's the chlorophyll. What does chlorophyll do? It absorbs light. What is the color of chlorophyll? It's green. What makes all the plants green on earth? It's chlorophyll. What do? They absorb light. What does light do? Light splits water. What is the splitting of water? What is it called? Learners, you need to know this term, that the splitting of water, when you split water into hydrogen and oxygen, you call that term photolysis. The word lysis means splitting, and the word photo means light. So we use light to split water to give us oxygen. And that is why we say photosynthesis. What is photosynthesis? Light. We use light to synthesize oxygen, to make oxygen, and we use light to make carbohydrates or food. And that, learners, brings us to an end on the factors affecting the rate of photosynthesis. See you next week for another lesson on photosynthesis. Study well and use your equations to understand photosynthesis. Use your schematic representations of your diagrams, know your labels and what happens in each part. Like the dark phase occurs in the actual lamella or grana. The light phase occurs in the lamella and grana, and the dark phase occurs in your stroma. Please know those facts and be prepared for your exam. Thank you.